Good afternoon, welcome on this third webinar in a series of 6 focusing on the work we've done in Galaxy on the COVID-19 data. This webinar series is hosted by Elixir, and today we will be talking about supporting the COVID-19 data portal, the viral data cleaning from human reads and submission to ENA. I'm Frederick Coppens from the VIB and head of Node for Elixir Belgium, and together with Bert, Ignacio and Miguel from the Elixir Belgium team, I'll guide you through the work that we've been doing to facilitate submission to ENA. Let's dive into the topic of today, submission to ENA and the necessary pre-processing to enable this. So I will start with a brief outline of the COVID-19 data portal, and then Miguel will talk about the submission of the viral data that we have been integrating. Ignacio will focus on the Galaxy workflows, and Bert will explain how you can deploy this or use these tools yourself in different contexts. So the COVID-19 data platform is a collaboration at the European level, which was coordinated by the European Commission and focuses around a platform that EMBL EBI has set up on top of the existing databases that they are running. This is a very broad and close collaboration with many people. So the European research infrastructures are heavily involved to provide data. Elixir is there coordinating a lot for the molecular side. And also a number of the international initiatives that collect data and provide analysis are involved in this, and the national infrastructures that are being built to tackle COVID are instrumental to make sure that we capture all the data and we have the necessary use cases. On the right, you can see a number of the projects that are funding this work in the different parties. The portal itself you can find on the URL at the bottom, and this collects much data across different disciplines related to COVID. And this builds on the databases that are already available, many at EBI, but also beyond. And the COVID-19 data portal gives a specific view on these databases related to COVID. So here you can find all the data related to COVID in one place, rather than having to explore all these different databases. And this data is becoming quite diverse, from viral sequences in human data to expression analysis, proteomics, but also now biochemistry, some imaging, and of course the literature that is exploded around COVID research. Today we will focus on the viral sequences part, and this is one small part of a larger project. If you want more details around this infrastructure, you can look on the webinar that we've done last week, which is already available on YouTube, where we explain the whole open infrastructure that we've built, where we connect the data portal with Galaxy instances and use these for analysis according to the standards that we've been building in different projects. This is a truly global collaboration, but today I will focus on the connection between the COVID-19 data portal and Galaxy, mainly on how we've leveraged Galaxy to facilitate submission of viral sequences to the European Nucleotide Archive as part of the COVID-19 data portal. So why did we focus on this? As a part of the COVID-19 data portal, the European Commission also encouraged all the countries to set up national data coordination teams. These are crucial to get the national labs and all the parties involved in a coordinated way to provide data and to provide input on what is needed to establish at the European level. And in the monthly meetings that we have, it was very early evident that access to data is really a common challenge. A lot of data is being produced, but getting this data publicly available is a challenge because these workflows to do so aren't necessarily set up. So the observation is that GISAID has a lot of the submissions because this is already part of what many clinical labs do, for example, for the regular flu. Submission to ENA, on the other hand, is something that's not commonly implemented, but this allows the public availability of the data. And so we really set out to get more and as high quality data as possible into ENA. And now we'll go a bit more in detail on how we did this, and I give the word to Miguel, who will talk about the submission of the viral data.
Thanks, Frederick. Yeah, so a bit more of a specific context to this issue. The GISAID is it's a repository for flu data, as Frederick mentioned. It is a de facto established repository for clinical labs and for virology labs. And at the heart of it is the database, which is for SARS, it's been named EpiCov, but it's based on the previous one, which is named EpiFlu. And they also have tools, analysis and links to tools to do mainly in terms of nucleotide sequences. They do phylogenetics, clade nomenclature assignment, and they do temporal and geographical distribution of the new clades. The database itself indexes consensus sequences only, so the data in the raw reads is lost, and some basic metadata on patients, collection information, and sequencing technology. Additionally to this, the database, it's not open, not fully open. It's accessible through registration after application, and it has a user agreement with limits on data reuse. In contrast, ENA, as you probably know, it's fully open. It has a more rigorous structured metadata model to accompany the data. It takes not only consensus sequence, but all types of genetic sequences, such as the raw reads, consensus sequences, and genes, but it's also linked to the IN SDC, DC, which basically means that you get access to NCBI and other nucleotide databases. And it's already at some level integrated with Galaxy in the sense that you can retrieve data from all these databases for further analysis within Galaxy. So our observation, what really started this, was we looked at who was submitting SARS sequence data to GISAID in Belgium, and we found that all the sequences, 100% were going to GISAID, but none were going to ENA. And then if you look at the world situation, it's a bit better, of course, but you still have many more sequences going to GISAID than to ENA. So you can have the next slide, please. A lot of these labs are producing lots of sequences, and for submission of bulk data sets is of course much easier if you do programmatic submissions, but this type of submissions, programmatic to ENA, do require some level of bioinformatics skills, which may not be necessarily present in clinical labs or virology labs. And so we thought that in order to increase the number of SARS-CoV-2 submissions to ENA, we needed to lower this technical barrier for bulk submissions. And we decided to do this by integrating existing analysis and submission tools into a more user-friendly environment, and we chose Galaxy. And we also did a bit of work on the metadata to make it, again, a bit more user-friendly. So what can you do with this tool? Well, the most basic thing is you can do, the submission is just one step. So there is no pre-registration of the study or the sample before the submission of the data and the metadata. So from the user's perspective, this happens all at the same time. The Galaxy platform will manage the ENA Webin credentials with the users, with the Galaxy user profile. We have also integrated an existing human reads cleaning tool, which is recommended by the ENA. So why do you need to do this? Well, human traces in viral data are considered human identifiable data. This is, of course, regulated by the GDPR, and it's also a requirement by ENA that these are removed before submission. And in terms of metadata, the tools, the metadata follows the ENA model, specifically the viral checklist for submission. And we offer different ways of submitting the metadata, either interactively or using templates that we've put together with the control vocabulary. Maybe we can go to the next slide, Frederick. So at the center of all this tool is the ENA upload tool, which is a command line tool developed by the Björn Gruning group at Freiburg. This does the bulk of the work. So this does the submission of the raw reads, and it uses tabular formatted metadata, which in turn converts to XML formatted data, which is what ENA takes. So the flow of the data is the raw reads can be cleaned if necessary to remove human traces, and then they're ready for submission. Metadata, as I mentioned before, can be input via a spreadsheet or interactively, and the tool will, with this information, make the tabular metadata files needed for submission. Data is transferred to ENA, to the FTP server of the user, and sample and study are registered. The accession numbers retrieved, and the rest of the metadata submitted to ENA, and that's basically a complete submission process. As I mentioned before, we identified lots of labs by going through the GIS8 submissions that were generating SARS-CoV-2 sequences and in Belgium, and we reach out to them explaining this the situation and offering to broker submissions of the raw data to ENA. And for this, we created a brokering account, and we managed to do so far 2 submissions or resubmissions from 2 different labs here in Belgium. Data from the University Hospital of Riverenland was supplied to us by the Institute of Tropical Medicine, and also data from Erasmus Hospital in Brussels was supplied to us by the Free University of Brussels. And we use the tool that we have with brokering account to submit them to ENA. And so far in the COVID-19 data portal, this is the only Belgian raw reads submission to be found. So I think next comes Ignacio, and he'll talk about the integration of the Galaxy workflows.
Yeah, thank you, Miguel. So can we go to the first slide? Yeah. So before actually going to the details of the technical details of the work, I just want to mention some of the general aspects of Galaxy and why we choose to use Galaxy. So we use Galaxy for many things, locally for bioinformatics analysis, but in this case that we need to deploy specific set of tools, we actually chose Galaxy because so it's a mature platform and it has a quite fast development and clear cycle of development. So that means that you can count on it for a long term. And also for some cases in our development, we need to get new features integrated. And with this fast development, you can count on it that it will be integrated in the next cycle of development. So you can count on it for the next version. Also, some things that are usually looked up but are important when deploying a set of tools and you want to deploy it for a specific set of users is that Galaxy already provides users management through different systems. It also provides reference data So that means that, for example, if you require a tool that will use a reference genome, for example, in our case, I will show later, but we are using it to map this to a human genome, you can count on it that it will be included in the platform. So that means that for the user, it will be much easier to run any kind of these tools. Also, it's important that it works with multiple infrastructure and also allows for the, different deployment options. So we use cloud mainly in our instance, but you can also use commercial clouds if it's like something temporary. Yeah, mainly because you have to pay for it. But you can also connect it to different HPC queuing systems. So that means that, for example, some tools that do require certain like more strict infrastructure requirements because they're more computational intensive, you can just by default, let's say for free with the installation of Galaxy, you can count on it. It's also possible to run it on a personal computer, and that's important because we want to include all of the possible users. So even those users that are, for example, in small hospitals or something like that, where they do have a bunch of data, but they don't have any kind of platforms, even though we provide an alternative solution, we also want to include them. And even if they only have a personal computer, they can also run it. So Bert will talk more about deployment later. Then it's also important that we are creating a set of tools and we are distributing a set of tools, but this is part of a bigger ecosystem of tools and workflows. So I will mention later, and it was also mentioned before in different webinars, that there are already available a lot of workflows to make analysis with COVID data, not just variant calling, but assemblies and phylogenetics. And now we also include the read, clean, and submission. But yeah, by putting things in Galaxy, you can count that they will live, I mean, the tool will live in this ecosystem. So in the next slide, I also want to mention some things that why we decided to use it for this specific solution that we are packing. So when talking about COVID data, Galaxy, first of all, it looks like the right way to put this tool, mainly because for a long time Galaxy has been like a platform that supports open data, and it's not just like a phrase. It's really, if you go into Galaxy, you can already use data from ENA. You can already do analysis from it, and I will show some examples later. But it was clear that it was the right place to put it because if you can already fetch data from ENA, it makes sense to actually already be able to submit data to ENA too. It's also important that, so Miguel mentioned before that one of our goals is to really lower the barrier and include everyone, any kind of users that can have data and want to submit it, to make that final step in the submission really easy. So with Galaxy, you can have a really user-friendly environment. When I'm talking about just a web interface, which is filling a form, which I will show later also. So that makes it really easy to create this kind of interface. Yeah, like together with this, of course, you will need any kind of computer science skills. It's just for most of the users, the entry point is just a web interface. It's also important that, yeah, as I mentioned before, some of the tools may require special computer resources, which not everybody has. So since this environment is available in different European countries and different Elixir nodes for free, then it's important that, yeah, that's a big feature of Galaxy, because then you can count that they will have a place to submit the data. And lastly, yeah, it's a production grade service. That means that I always say that one of the first problems that users encounter when running tools is actually installing it. So with these kind of systems, well, in many cases you don't really need to install anything, but in case you need to install it or run it, what we call a production grade is something that you wouldn't get errors right away. So that's more or less the features that we took into account when deciding to use Galaxy. And now, yeah, again, this is the overview that Miguel mentioned. in this case, it's already included which parts are actually part of the Galaxy with things we put into Galaxy. As Miguel mentioned before, we have two main steps. One is to remove the human reads, and the other one is actually the upload tool. So Miguel already mentioned more or less what are the inputs. The most important, yeah, just one thing. The most important thing here is that in the end, we're going to be doing the ENA submission, of course, and that requires the handling credentials for it, because yeah, it's an external service. It has different kinds of credentials besides what is included in a Galaxy instance. So that's an important step that Galaxy allows us to do. Now to the next one, please. So going to the details of the first step, I mentioned it was, so Miguel actually mentioned that it was a big requirement for us to remove the human reads, to not leave any kind of trace before submitting to a public repository. So removing the human reads is actually quite standard process when you do an analysis on viral data, because of course you only want the reads that belong to a viral genome. In our case, we have a clear goal, which is submitted data to ENA. So what we did in the first place was actually contact ENA and ask them, okay, what's the easiest method or the most standard method to actually do this pre-processing, not just to clean the human reads, but also for the quality control. So they pointed us this workflow which is maintained by the FinLab. You can see the second link. It's actually a bash script. It uses quite common tools, so it was not a problem for us to convert it into a Galaxy workflow. Just to go over it very quickly, it uses string galore with mainly standard parameters to do the quality control. Then BWA is used to map to the human genome. As I mentioned before, when distributing the tool with the Galaxy, you can count on the human genome being available, which is quite important, because if you want the users to first fetch the genome, make the index for the tool or something like that, then you are already losing half of the people. So BWA, as I said, it actually maps the read to the human genome. Then what you want to do is to keep based on the header, you just filter the ones that are actually mapping, and you keep only the ones that don't map to the human genome. And the last step is just to convert back to the original file format, because ENA has a specific subset of file formats that it takes, and you need to have the files in that format. Next one, please. So yeah, that was the workflow in general, but we actually went a bit far with it. As I mentioned before, one big requirement we have is actually making it super easy for users. And our experience tells us that having a single tool with single form is definitely easier. So in this case, what we did is actually pack the whole workflow in one single tool. So in this case, the users don't really need to do much of it. So they just select which are the input that they have, which is like the raw reads. This is actually a real example. So that's why I needed to blur the data sets, because they belong to actual data we submitted. So this is the first step. So when you click execute, you instantly get the clean reads. And that for the user is I mean, for distributing a tool is quite a big step, because it makes it really easy to run this step without actually the users needing to know any of the details of what's going on. So in the next one, please, next slide. Yeah. So as I mentioned, this solution, I mean, this workflow that we use for pre-processing is kind of the best fit that we could find for the goal we have in mind, which is submitting data to ENA. But of course, different cases, different use cases can have different pre-processing workflows. So that depends, for example, if you have different kind of input data, if it was sequenced with a specific kind of tool, like nanopore tools, which have like longer reads and may require a specific mapper, or if you're going to use or to do a different kind of analysis later on, in that case, it makes sense to change the workflow, and Galaxy allows to do that. So there are other different approaches to do the pre-processing. You can, of course, retain the reads by just mapping to the viral genome, because that's actually what you want. And you can also combine the human and the viral genome, do the mapping, and only retain the viral reads. Most important part here is that these other alternatives are also publicly available, so you can interchange them. I'm talking about in your private deployment or somewhere else. So if you go to workflow.hub.eu, and I really encourage you to go there, you can find not just this one, the one that we use, but also you can find a lot of similar ones, and you can explore difference between them, and you can easily run them in Galaxy with your own data. So yeah, the goal of this, and what is really cool about this, is that you can interchange, and with this, with one single idea in mind, you can run different versions of a workflow. So that's it for the reading, the removal of human reads. So again, we are now at the point that we already have the raw data with the cleaning step done. Yeah, indeed, where the pointer is pointing to. So we have all the input data for the ENA upload tool. So the next step, in the next slide, I'm focusing on the upload itself. So as it was mentioned before several times, we are reusing a tool that was developed in the group of Björn. So this tool actually takes care of the really, really low level details, which is connecting to the ENA server, and also making sure that the data fits the ENA defined schemas, which are definition of how the XML file needs to be structured, and also the languages that need to be used for a submission. So that's already something that for sure the user will need to take into account, because yeah, it's completely transparent for the user. So what we did on top of this is to actually create a wrapper that it does several things, but I just want to highlight the most important one. So first of all, it provides the user with an interactive graphical user interface, and it also provides the option to use spreadsheet templates, which again may not be the best practice, but in our experience, it's really necessary if you want to include everyone and every kind of user, and you really want to promote the submission of data. Then another thing that is important is that it associates all the information that it's being provided by the user with the data model that ENA uses, because ENA uses a very specific data model which consists of samples, run experiments, and studies, and these structures needs to be maintained if you want to make a submission. Again, for the user, by just filling a form that I will show later, you get all this mapping for free. Then it's also important that we are dealing with data that needs to be kept in private. So it's common that, for example, hospitals, when they do the sequencing, they name the samples with IDs that are private. So the tool also takes care of renaming these and creating Unix IDs which have no sensitive information. So they are also not just the reads itself, but also the names of the data files have no trace information. And the last one, as I mentioned before, since we are dealing with public repository, we have external credentials. And so the tool, on top of what Galaxy does, needs to take care of the credentials that maybe the user is providing, or if it's like a system-wide credentials, for example, in the case of a brokering platform, the tool needs to take care of that, because the user doesn't know about the credentials that are being used by the brokering platform. So in the next one, please. This is the view that the user gets from the submission tool. As some of you may know about Galaxy. If not, in the middle panel, you have the form that you actually need to fill. As you can see, some things are already pre-filled with languages that ENA uses. And on the right side, you have the data sets. So you just fill the form with the metadata that is requested, and the tool takes care of matching that to the metadata format that ENA uses. And then you just click execute and the submission is done. Yeah, so now going back to the really big picture of it, this will be like this, what I mentioned before is like the solution for the submission step. But as you can already imagine, and of course, if you visited the previous webinars, there was a lot of explanation about different analysis that can be done with public data. So as Miguel mentioned before, one thing that we really want to get is to increase the number of submissions. And for that, we have two things to do. One is to lower the skill barrier that the users need to go through before doing the submission to make it really easy. But also, we think that it's important to involve the users in a different way. I mean, involve them in the whole pipeline of analysis. So by providing workflows that they can use, and also by providing results of workflows that other people already did and results that they got from public data, we are actually telling them, okay, look, if you submit the data, there is a lot that people can do with it. So this is actually a way, providing the workflows is actually a way of telling the people that the submission is important and they are actually contributing to this analysis. As well, of course, if you visit this website and also if you go to Workflow Hub, you can see a lot of analysis that can be done with your data. So yeah, it's actually part of the big picture and what we are showing the users to before they actually submit. And the last part, before I go over time. Yeah, so this is actually a bit of a promotion. In 2 weeks, there will be a webinar, part of this series. We will be talking about the viral beacon. So if some of you know about the beacon interface, it's actually a way to query metadata about datasets. It's in principle planned to use with clinical data, which should remain private. But in CRG, they already have a viral beacon, so you can use this viral beacon to query metadata about the genome of the viral data. So yeah, it's just to mention that, and if you want to come to this webinar, you probably don't want to lose it. And now, yes, that's would be it for me.
Thank you, Ignacio. So there are multiple ways in how you can deploy and use our Galaxy tool. And obviously, you need Galaxy for this. Galaxy can run inside a container. And this is a very easy way to run your own instance on your personal laptop. And if you have high computing demands, you can also deploy it on a cloud infrastructure. This container can also be used for brokering, as we will see later. Next to running it inside a container, you can, of course, also install the tool on a public instance where it can be used by everyone. Next slide, please. Containers are something that everyone by now probably already heard of, and it's gaining popularity by the day. Since a container contains all the libraries and dependencies, it is perfect for preservation and reproducibility. The most popular of these container systems is Docker, and it runs on most of the user operating systems, including Windows, Linux, and Mac OS. The container itself is built from an image, which can be seen as a blueprint of the container. And these images can be shared through a registry like Docker Hub or K.io, which makes it very easy to share. This means that if Docker is installed on the system, it is enough to run the container. And in this case, the container will have everything to run Galaxy. The lab of Björn Grünig built a container with like a bare bone Galaxy instance inside, which is for us then ready to use at only one command as seen here on the slide. Next. The barebone Galaxy container is, as the word explains itself, without any tools or workflows pre-installed. So installing the tools, workflows, and the data required for specific analysis inside the Galaxy container in an automatic way was actually already done for the tutorials in the Galaxy training network. This established system was used to create our own flavor Galaxy container containing our wrapped ENA upload tool. Next. So here we see again the overview, and with the tools that Ignacio showed us for cleaning reads and uploading data to ENA, running in Galaxy, and this then inside a container, which is then easy to distribute and deployable, as we will see in the next slide. So, or the slide after this one, but once you start the container, it will look something like this, with all the tools you need on the left, and in the middle panel, there are all the steps that you need to follow, and they will guide you through the submission. Next slide, please. So if Docker is installed, people only have to run one command, and then they will have the full suite available in their browser. And the ENA submission tool, of course, needs to know the credentials of the ENA Webin account in order to submit. In 2020, there was a possibility added to Galaxy itself to handle this sensitive information on a user or on a system-wide level. And in the container, user can just go to the user preferences and manage their information and can add their credentials for the ENA Webin account in a secure way, as seen on the slide. And running this container locally takes away GDPR worries that come with processing raw reads that still contain human reads. Next slide. So since the read cleaning is quite a computational demanding step, and running this container on your own laptop, this can give some troubles. Luckily, this ENA upload container is not only easy to deploy on a laptop, but also on a cloud infrastructure. And at Elixir Belgium, we could actually set up a Galaxy instance in no time using this container. And this instance is dedicated for Belgian researchers that want to submit COVID-19 data. It is deployed at covid19.usegalaxy.be for people that are interested. This is done on the Flemish supercomputer, which provides plenty of computing power for the computational analysis part, and also the network bandwidth that is needed to upload all the raw data. This instance also gives us the opportunity to set up credentials for an Elixir Belgium brokering account. And researchers then have access to this instance based on a request. So they can upload the data using the brokering account without the need for registering to ENA themselves. And in this scenario, the credentials are set up system-wide by the administrator, facilitating this process further for the researchers. So, and this centralization also means that we can give additional support during submission when needed. So, next slide, please. The ENA upload tool can also be deployed on a public Galaxy instance, and this is showcased on usegalaxy.eu, an instance that is available worldwide for everyone. And of course, we don't use, in this case, a general brokering account, but instead users will set up their credentials, like explained before, in the user information section. And this will soon become available on usegalaxy.be too. Next slide. The make submission of raw reads to ENA easy for everyone. A screencast was made, which explains every detail from deploying your own Galaxy container to having your data and metadata on ENA. So if you're interested, please check this out. This was it for me.
Thank you, Bert. So wrapping up, we've showed you today the work that we've been doing to further develop the ENA upload tool, wrap this inside Galaxy, combine this with workflows to pre-process the data to remove the human traces, and deploy this in a container and different options to submit the data then to ENA. So with these different solutions, we try to cover different use cases and different audiences. You can take just the command line tool and integrate this in your specific environment. If you have a management system already set up, you can integrate this and submit your data through this to ENA. You can deploy your own container on your local laptop, on your local infrastructure in your institute to take care of all the processing steps in a familiar and trusted environment. You can also use galaxy.eu or other public instances to make these data available and analyze them. And as mentioned, for Belgian researchers, we made a dedicated instance to provide additional support. Other people can easily take this container and run or provide a similar service if needed. The reason for limiting to Belgian research is just pure of bandwidth of the people involved on our side. There's a lot of things that we are planning that we want to work further on. So we've mentioned the different workflows on assembly, on variant calling, and so we want to integrate them into the container and the instance that we provide. We want to also enable submitting the consensus sequence, so the viral sequence, to ENA with all the required metadata. This is a high priority now. And if we have that, we want to explore the simultaneous submission to both ENA and GISA, that we can cover both with one go, because this is common practice and sometimes a requirement in certain countries. The sequencing data or the reads are one part. The result of downstream analysis are also interesting to integrate. And so, for example, inclusion of variant data and being able to submit this, for example, to the European Variant Archive is definitely also in scope later on. And the deeper integration with data management systems, we are locally using FairdomSeq for that, is also something that we are exploring together with the Fairdom consortium that we are involved in. And ultimately, we want to work towards an integrated and sustainable health research, healthcare ecosystem that seamlessly can distribute this data in a way that is GDPR compliant. That's a relatively ambitious goal, but now there is the momentum, so we can make good use of this current crisis to start establishing that and make progress here. As we mentioned last time, we really see this as an exemplar implementation of the European Open Science Cloud. And this is not just a European project. We've done this in a global context where we provide one part of the puzzle and many other partners are involved to come up with the whole ecosystem. And as was already mentioned several times, we're building on existing open infrastructure, on existing tools that we repurpose or we put some additional things on top to make them more user-friendly, but there's hardly any new developments from scratch to make this happen. The slides will be available later on. We've collected here some of the interesting URLs. In the first one, you can find all the information on this work with in the RDM hub that we've made for Elixir Belgium. Finally, I just want to acknowledge the funding. Elixir Belgium is supported by the Flemish Research Foundation and the Policy Office for Economy, Science and Innovation, as well as the EOS Live project. And these fundings were available before the COVID-19 crisis hit, and we built on the plans that we had there and reoriented them towards COVID. And as mentioned, it's really a collaborative effort, work that has been shown by the VIB and Elixir Belgium team was done in collaboration with the Freiburg team and the ENA and people at EBI to make this connection work. And this was a very productive and efficient collaboration. Compute resources are provided by the Flemish Supercomputing Center, and this features in the wider network. It's a fantastic environment to work in, and I really want to thank and acknowledge these people for the very nice collaboration.